All right, so what we're going to be talking about today is a process called signal transduction pathways. So technically, y'all have already been learning about these through the steps of cell communication, but we just now need to, go in a little bit more detail and learn some of the names of all of these steps. And so with signal transduction, okay, it's the same 3 steps that we already talked about. So we talked about reception. Okay, so reception from there goes to transduction. And from transduction, you have some sort of response. So we need to remember that when we're talking about these 3 steps, we're talking about very specific pathways. And so our receptors are very specific to certain ligands. Okay, so receptors specific to ligands. And remember, ligands are signaling molecules. So they can be like really small proteins, really small charged molecules, anything like that. And so what's going to happen is when that ligand binds to that receptor, there's going to be a conformational change, and then it's going to cause some sort of reaction. And so we have to remember that many signaling pathways include protein creation, so protein synthesis, protein modification, and phosphorylation. Okay, so responses include protein synthesis, / modification, and phosphorylation. Okay, so remember, phosphorylation is the addition of that phosphate group. Okay, so I think it's tomorrow on our calendar, we're going to be talking about some diseases and how they're going to interfere with these different types of pathways. So first, let's go ahead and talk about reception. Okay, so I'm going to scoot this on over. Remember, you could pause the video at any time. Okay. All right, so when we're talking about reception, we're talking about that ligand molecule physically binding to a receptor. Okay, so when the ligand binds, there's a conformational change. Okay, so conformational means like a change in that shape. Kind of like how enzymes work. Okay, so with that conformational change, that's the initial transduction. Okay, so this is what allows that next step to happen. Okay, so I have my little ligand here. It's going to bind to a receptor. Okay. There's my receptor. Okay, and this receptor right here is going to change according to the shape of that ligand. And so I can have 2 types of receptors. So I can have intracellular receptors. Okay, so remember, intracellular means inside the cell, so this is going to be something that's in the plasma membrane. And so because of that, that ligand needs to be able to enter into the cell membrane, so it has to be really, really small, or it has to be hydrophobic. That's the only way that the ligand is going to enter into that cytoplasm. And then, of course, we can have receptors on the plasma membrane. Okay, so these receptors are really what we're going to be focusing on today. I have actually 3 examples that I want to give you, just so that way you can understand how a receptor is going to work and maybe what they're going to look like during this process. So let's go over 3 different types of receptors. Our first one is a G protein receptor. Okay, so G protein receptor. Don't get intimidated by this, all right? So what this is a very basic receptor. So I can see on the outside of my cell membrane, I have this little G protein receptor. And I can see underneath my membrane, I actually have a G protein. And I know this picture is really fuzzy. So what ends up happening is my signal molecule is going to bind to this receptor. when that happens, that signals GTP. So GTP is an energy molecule. We haven't gone over that yet, but it sounds just like ATP. So GTP is just like that. It's an energy molecule, and so what's going to happen is it's going to energize this signal to activate my G protein. That G protein, because it has energy on it, is going to travel over to the protein or the enzyme that it needs to activate. So this is almost like a multi-step. So this is a membrane receptor that works with the help of the G protein. With help of G protein. I don't think I have enough room here. Okay, so it's a membrane receptor that works with the help of the G protein. Okay, so once my ligand binds, okay, so my ligand's going to bind right here, it's going to activate this G protein by giving it GTP. The GTP will bind to that G protein and it's going to move. All right, so I'm going to put right here, ligand binds and causes G protein to activate and let's see here, activate cellular response. That's probably the best way to put it. Okay, and in this picture, the cellular response is activating this enzyme. Okay, so that's what a G protein does. G protein, you have this little G protein underneath the cell membrane. Once the receptor is activated, that G protein will become activated, and now that it's activated, it's going to come over here and activate another protein, an enzyme, whatever it is. So up here, okay, when there's no ligand, that G protein is inactive. All right. So this is for one cellular response. So I can see right here, I only have one cellular response from that G protein. Sometimes our body needs multiple responses. And so our next receptor, okay, it's kind of scary looking. Okay, it's a tyrosine kinase. Okay, and let's see if I can fit it all on the screen. Okay, so it's kind of scary looking, but essentially what this is, okay, this is a receptor, and both receptors take the same ligand, but both spots have to be filled. So if I have a ligand and it adheres to both of these receptors, it's going to cause all of these right here. These are called the tyrosine kinase protein receptors. It's going to cause all of these to become phosphorylated. So I can see I have 6 ATP coming through, and it's going to phosphorylate all these molecules. So I can see right here, okay, it's inactive because it's open. Once my ligand binds right here, it's going to become active and it's going to close. And so I have 6 ATP molecules coming around, and because I have 6 ATP, that's much more energy. I can have a lot more responses. And so this is different because it allows for multiple cellular responses. Okay. So the ligand binds to 2 receptors . Okay, and activates tyrosine kinase. Okay, so this allows for phosphorylation. Okay, essentially allows a battery to charge it. And from there, initiates several responses. Okay, so that's the difference between a G protein and my tyrosine kinase. Okay, so remember, same ligand. Okay, so not the same ligand as over here. Okay, so these are 2 different ligands. Okay, so this is a ligand that's specific to my tyrosine kinase receptor, but I have two of them. Once they're activated, they're going to close. Okay, this is called a dimer. They're going to close, they're going to become phosphorylated, and they're going to be producing several responses. Okay, so that's the difference between those two. And then the last one, okay, last one's very short and simple. Okay, so we're going to talk about this one a little bit more when we talk about some of our body systems, but I don't have a picture of this one just because it's very easy. So when we have a ligand-gated ion channel, this is when you have specific ligands that cause the channels to open and close, and it regulates flow of ions. Okay. So ligands cause channels to open and close to regulate ions. Okay, so channels like, for example, in our nervous system, okay, that's where you're going to find these ligand-gated ion channels. Okay, so this is specific towards ions. I'm going to underline that. Okay, specific towards ions. All right, so next, okay, we're on our next step of transduction. This is our middle step. We've already talked about reception. Now we're on transduction. So transduction, we've already had that ligand stimulate our receptor, and so we need to go through the process of creating a response. And so during this step, we can have multiple molecular interactions. So we can have multiple signals to multiple target molecules. Okay, so we call this a cascade. Okay, so a cascade of molecular interactions to relay signals. Okay, so remember, okay, cascade, think of like a waterfall. Okay, that's a cascade. So you have molecular interactions in order to relay signals. All right, so during this time, we can amplify our signal. So sometimes we have something called a phosphorylation cascade. Okay, so I'm going to bubble this right here. Okay, so a phosphorylation cascade. I can see, okay, here's my receptor with my signal molecule. I have an inactive protein kinase. Okay, so I'm going to put a little star right here. Kinases, okay, amplifies signals. Okay, so if you see a kinase here, that means that that signal needs to get bigger. Okay, so I have an inactive protein kinase here. It's been activated because of my ligand bound to my receptor, and so it's activated my relay molecule. This is going to happen over and over and over again. And so this happens because I have ATP available. And so now I've been able to amplify this. This is only one protein kinase, but I can have multiple. Okay, and I can see right here, there's another protein kinase 2, another one 3. I can have multiple of these. And so what this does is it greatly amplifies my signal. So this is called a phosphorylation cascade. Okay, so a phosphorylation cascade will amplify my signal. Okay, phosphorylation cascade amplifies my signal. Okay, I feel like I'm writing really bad today. All right. So during this time, I can also have secondary messengers. Okay, so secondary messengers can also initiate that phosphorylation cascade. Okay, so what does that mean by a secondary messenger? So a secondary messenger is going to be something that's inside my cell membrane already. So once I have my receptor here, the signal molecule, okay, so here's an activated relay molecule. Okay, so this relay molecule could be my secondary messenger. I had to have my protein kinase activate in order to activate that relay molecule. Okay, so I'm going to put right here, secondary messenger. I'm going to abbreviate. Okay, secondary messenger. And so the reason why I wanted to bring that up is because later we're going to talk about some secondary messengers such as cyclic AMP. Okay, so cyclic AMP, I'm just throwing this out there for y'all just to become familiar with that word, is a type of secondary messenger. Also in our nervous system, calcium ions. Calcium ions are secondary messengers. Okay, so from here, after transduction, we have some sort of response. Okay, so usually our response can be a variety of things. Okay, so it could be regulation of transcription, or cytoplasmic activities, okay, so when that happens, okay, genes are turned on or off. Okay, so for example, okay, genes can be turned on or off. All right, and then of course we can also modify proteins. All right, so something that we're going to talk about a little bit later is going to be apoptosis. So we're going to talk about apoptosis more when we talk about genetics. But when we're talking about apoptosis, that's going to be programmed. Essentially, we define that as programmed cell death or programmed cell suicide. But it's very much on purpose. So, for example, in utero, okay, so we actually all have webbing between our fingers. Okay, and that's why we actually have a little bit of webbing right here at the bottom. And so during fetal development, we actually have a step where our genes are activated, so that way that webbing goes through apoptosis. And so if we're talking about evolutionarily, that's why things like fish, frogs, things like that still have the webbing in between their toes. And that's just because, apoptosis did not turn on for those particular animals. For us, it does turn on. And sometimes you do have a mutation where that gene never activates, and so you'll have a baby born with webbing in between their fingers or their toes. But we're going to talk about that later when we talk about genetic mutations. So go ahead, copy down these notes. Let me know if you have any questions, and I will see y'all later.